Hello, everyone, and thank you for joining us today. And good morning to our guests from California that joined us so early in the morning. My name is Ifat Adaya, and I work at the International Division of the Israel Innovation Authority. And I look forward to this valuable webinar with our friends from Lawrence Berkeley National Laboratory, funded by the US Department of Energy. I would like to invite Tammy Warshevsky, who heads the business development efforts in America and Europe at the Israel Innovation Authority, to share some opening remarks. Tammy, the floor is yours.
Thank you, Ifat. I see that participants are still joining. So they will have this time before we start with the content. So good afternoon in Israel, good morning in the US, and welcome to this special webinar that we hold together with Berkeley National Labs. This is in fact part of wider collaboration the Israel Innovation Authority has with the DOE for many years, collaboration that we value very much. And basically, this event reflects maybe in the best way the great value of global collaboration as we see it. First, because it lies on shared mission of both organizations to turn scientific breakthrough into real-world application and impact. And second, because it brings the best value of both sides. When we talk about innovation, the scientific discovery, it's only the start. But the real challenge is actually about navigating the full journey from the scientific complexity through the market access and integration towards the challenge of scaling. These are not easy steps, and they rarely happen in isolation. They require collaboration across disciplines, they require collaboration across sectors, and definitely across borders and countries. And that's why international partnership like this is so critical. Berkeley Labs, you will hear very soon, brings world-class capabilities in biotechnology, bioenergy, as well as in scaling disruptive technology. Israel ecosystem brings deep tech expertise, strong culture of entrepreneurship, and agile innovators. And together, basically, these are complementary capabilities. And so together we can definitely move both faster and also reach higher. So with this webinar, we will unfold opportunities on both joint R&D for scaling disruptive technologies and for knowledge commercialization from the labs. Alongside, we will share the funding opportunities that offered by Israeli Innovation Authority to support such collaboration and make it easy for everybody to move forward. So thank you, Deepti Tanjur and Robin Johnston from Berkeley Labs for your availability to be with us and share with us this great opportunity today. And for Aviv Ze'evi, VP of the Infrastructure Division at the Israel Innovation Authority. And with this, I wish all of you good luck, and hopefully a fruitful collaboration will unfold from that.
Thank you, Tammy. And thank you again, Robin Johnson and Deepti Tanjor, for joining us today. Before Robin presents the collaboration opportunity on knowledge commercialization, I would like to invite Dr. Aviv Ze'evi, VP of Infrastructure Technologies at the Israel Innovation Authority, to give some context to our incentive program, which can be complementary by supporting the validation process. Aviv, over to you.
Thank you so much, Ifat, and thank you all for joining us. And I will try to explain exactly how can you collaborate, which means which is the relevant incentive program that can help you to collaborate and to be funded for this kind of collaboration. So we are calling it the Knowledge Transfer and Commercialization Program, which is part of the relevant program of the research in the industry of the Israeli Innovation Authority, part of the research fund that we are managing here in the Infrastructure Division in the Israel Innovation Authority. So the goals of this specific program is to validate a specific IP that was developed by a foreign academy, in this case, the Berkeley University or Labs, which has developed the IP. We are talking about a relevant, mature IP, which can be relevant for commercialization by an Israeli company. So we are validating this specific IP by, for instance, using a lab that we have developed in the Israeli industry, and we are trying to adjust the relevant IP to our needs. So we want to make sure that the IP is working fine outside of the lab and not only by using the equipment of the relevant lab. And the project itself that you are submitting to us will be mainly focused on the validation, the adjust, and the implementation, which means a platform for the economic potential of the knowledge. Let's take an example. Let's say that the Berkeley labs have developed a specific algorithm, and the algorithm is a way for optimizing a specific process. You want to validate that this specific algorithm is relevant for the relevant data that you have of the Israeli company. You want to adjust the algorithm in case there are some parameters that still needs to be developed. And in some cases, you will implement specific algorithm on your data and make sure it works fine even before you start to develop based on this as a specific service or a product. So the term, the motivation itself is that the knowledge commercialization program is focused on creating some kind of collaboration. There is an IP was developed by the academic partners. There is an Israeli industry who is willing to use it. We don't ask for any exclusivity of the IP. It means that the Berkeley labs may, of course, use this IP. They still own the IP, and they can commercialize it also to an American company or any other company around the world. But you have to make sure that it's logical for you to use this specific IP under the terms that you have discussed with the Berkeley labs. The main idea is to close the gap. It means that the IP that was developed by Berkeley still might not be relevant for industrial use. Maybe the protocol is not good enough. Maybe it lacks specific parameters, and you have to make sure that it's relevant for you. And the lab, the Berkeley lab, will be served as a subcontractor of the Israeli company, which will submit a proposal to the Israeli Innovation Authority in order to be funded. So the terms are that the IP or the entire project has to work on TRL 4 and 5, technology readiness level. The duration is up to 3 years, and the financing is up to 60% of the approved budget. It means that you are submitting a specific budget, both of the Israeli company and the Berkeley labs. We make sure that the relevant part of the Berkeley lab is up to 50% of the project budget. So it means you have to share the tasks between you two. So you are you're submitting a proposal, we approve it, the application itself. We make sure that the budget is relevant to the costs that are eligible, and we are paying 60% of the approved budget. You may spend, as I said, up to 50% of the budget on the foreign academy, which means that the Berkeley labs may get 100% of what they need, but we will refund up to 60% of the budget. We already have around 10 to 15 projects per year, which we have already funded between an Israeli company and a foreign academy, with Australia, Austria, Germany, CERN Institute in Switzerland, Princeton University, etc. So we already know it's working, and we have, I would say, some customer or clients that are really pleased from this kind of cooperation. We are aware of the fact that there is a very good academia in Israel, but we are not excellent in everything. There are some small parts of the world, like the US, which is able to teach us what is really a good IP in some areas. So how do you apply? What's the process? So you should sign, before you submit a proposal, an MOU between the two parties, a detailed definition of the IP itself, which means what exactly is the IP. You have to, for instance, depends if the IP is relevant for a specific sector, is it patent, who owns it, for how many years will you get the license, etc. What are the costs and the terms, which means how much will you pay to the Berkeley? It might be a down payment or maybe royalties or both. This is all up to you, and we are not involved in the terms themselves. We just want to make sure that after we evaluate the proposal and we approve it, you can already start working, and it doesn't take another 2 to 3 years before you get to a decision of how to share what is the real cost. And of course, you have to decide on the responsibilities, which mean who does what. What are the relevant tasks will be done by the local industry or the Berkeley labs. And as I said before, the eligible tasks are validation, IP adaptation for the company needs, training, of course, because maybe the Israeli R&D team needs to learn to be trained about a specific IP and how to continue the development of this specific IP. So the application needs to be submitted, as I said, only by the Israeli company. They are negotiation with the Berkeley labs. The application forms, including appendix of the budget itself, is a Word form, includes the detailed request, an Excel form which includes the detailed budget, and an Excel form which includes the detailed budget for the Berkeley relevant lab, which means what is the equipment, the salaries, the materials, etc. And that's exactly, I would say, all I have to say. The relevant person who managed this specific program, his name is Rami Tzafon. He couldn't speak today because he's now representing us in the OECD. But you have his mail here, you have his telephone number in the office, and you can contact him. And those of you have already identified a few names, you can also contact me directly, and I will be really happy to help. Thank you very much.
Thank you, Aviv. And now I would like to invite Robin Johnson, Director of Commercialization at Joint Bioenergy Institute, led by Lorn's Berkeley National Lab, to speak about commercialization opportunity. Robin, the floor is yours.
Okay, let me just pull up my slides. Hi, everyone. Thanks for joining this morning. Let me get back to the, here we go. Let me get back to the very front. So I'm Robin Johnston. I'm the Director of Commercialization for the Joint Bioenergy Institute, which is a Department of Energy funded institute. And today here's the outline. I'm gonna give you the background on JBay, talk about our mission, our IP metrics, so you can see that we do produce a lot of IP. We'll talk about industry collaborations and licensing and startups. And then we'll have, I don't know, are we going to save the Q&A for the end?
Yes, we will save the Q&A for the end. Please just put your questions in the Q&A tab and we will address them at the end of the webinar.
Sounds good. Okay, so here's the background on JBay. We are a collaboration of 5 U.S. Department of Energy national laboratories and 9 universities across the country. And here's the map of all of our partners. And 95% of the people at least are at JBay a majority of the time, but 85% of the FTEs are working at JBay almost 100% of the time. And this is just a little outline of of our lab space. And this makes it very easy for JBay to coordinate on IP management. Berkeley Lab manages all the IP that comes out of JBay, regardless of the institution that created the IP. All those individual partners still have ownership of their IP, but it's all managed by me, basically. A lot of coordination goes into that. So our mission is to establish the scientific knowledge and new technologies to transform the maximum amount of carbon available in bioenergy crops, and that's all the lignocellulosic components, into advanced biofuels and bioproducts. We do not work on ethanol, but we work on all other kinds of biofuels and bioproducts. JBay has 5 different divisions. I'll start with the middle row with feedstocks. We do feedstock engineering. We try to reduce the amount of lignin or tailor the kind of lignin that plants are producing so that the plants are more easily deconstructed into their components. We have several strategies for doing that deconstruction and treatment. One is that we take the lignin, which is the polymer that holds the polysaccharides in the fibers of the plant, and we can extract the lignin first and then convert that and valorize that, and then take the polysaccharides and feed them, and that's C5 and C6 sugars, into microbes that have been engineered to use those polysaccharides. Or we can actually depolymerize the lignin, feed it with the polysaccharides to the microbes, and many of them can use aromatic C5 and C6 sugars. We're mostly working on the host organisms Rhodosporidium tortuloides and Pseudomonas putida. We do some work in E. coli and yeast, but that's more fundamental work before we move into those other hosts. Then we take those microbes have also been engineered to produce a variety of different biofuels and bioproducts, and I'm sure that you all are familiar with companies that are doing that right now. We also have a life cycle economics and agronomy division called LEAD. We do a lot of techno-economic diligence on our technologies to decide which ones to push forward into the pipeline. And that's fairly rare, even for universities to do that. So we're kind of proud of the fact that we do that. We also have a technology division that supports all of those other areas. Here's just a snapshot of the IP landscape that we've developed over the past 18 years at JBay. As you can see, we have a lot of IP. We've done a lot of licensing. So that's just to show you kind of our track record. So how do you collaborate with us? There are 2 partnering mechanisms. One is called a cooperative research and development agreement, and that is what it says it is. It's very cooperative. We collaborate with research on with the industry partner for shared benefit. So it's something that Berkeley Labs should be interested in doing, sort of pushing forward our frontiers as well as responding to industry questions and pain points. In this type of arrangement, both of the parties have tasks that are collaboratively developed, and we develop a statement of work together. Under a CRADA, there must be in-kind support from the industry partner. That means you have employees working on the project, or you're supplying equipment, things like that. The one great advantage of a CRADA is that you can actually send your employees to be embedded at JBay to work side by side with our researchers. And several companies in the past have used that mechanism to train employees in the cutting edge areas of synthetic biology and plant engineering and things like that. So that's been very successful. Another way to work with us is through a strategic partnership project agreement. That's basically more like a work for others, where you have a defined scope of something that you wanted us to do for you, and it's less collaborative. We're also happy to do that, and as Deepti will tell you, they mostly do SPPs, I believe. This is typically used when you have the background IP or expertise in the product that you want to make, but we have some kind of techniques, proteomics, scale up, things like that that you would like to leverage. We do have, and I was telling our hosts that we do have a U.S. manufacturing requirement for our CRADA agreements and in our licenses. So I wanted to just get this out up front. Basically, both the CRADAs and the licenses, even if there's a non-exclusive license for patent rights, not for software, but for patent rights, the companies must substantially manufacture in the U.S. This used to say substantially manufacture in the U.S. for the U.S. market, but that has been changed in the last couple of years to say that you must substantially manufacture in the US for the global market. Now, it's possible in the past that you could have gotten a waiver to this. We have not seen waivers recently, but it doesn't mean it can't be done. Here's just some examples of partnerships we've had. We've had SPP with Mitsubishi working on one of our fuel molecules, our energy-dense fuel molecules, and putting that into yeast. We've worked with Novozymes under a DOE-funded CRADA, a startup company called Protein Evolution that's working on plastics recycling and enzyme development, and we did a CRADA with them. One of our big success stories is a 10-year, $3 million partnership with Total Energies. They embedded employees at JBay for, I think it was like 8 years or something. We trained 4 of their employees, Angelique Chanel, who's now the biofuels R&D lead for Total Energies, and Florence Mingarden, who does R&D at Total. So that was a very tight collaboration, very productive. We also had a collaboration with a company called Verdia. They wanted us to validate some of their processes. We did that under an SPP and then worked under a CRADA for new technology. They were able to use that data to when they presented to Storo Enzo when they wanted to seek out an M&A, and they were successfully purchased. Another way that we interact with industry is just straight licenses. And we have licenses with Futurigene. They are working on developing some increased cellulose content technologies, and they're working in eucalyptus, and they've gone to field trials with some of their traits, but not the JBay trait is still earlier in their pipeline. We have software licenses with Digitalis, Altos, and CJ. So those are all sort of the tools that we've licensed. One of the important ways that we get our technologies out and used by companies is by creating spin-outs and startups, because we do, most of our technologies are TRL level 2, 3, maybe 4. And this is a way to bridge between our early stage research and industry. So we typically have an average of a startup company a year. This past year, we had 2. And I would encourage you, if you're looking for high TRL level technologies, to partner with our startups. That's what they are there for, to partner with industry to get our technologies out and used in the broadest way possible. So just as a little aside, we have lots of programs to encourage startups. And one is these fireside chats with previous JBay entrepreneurs. Here's 2 examples here. We just have informal gatherings so that JBay researchers can get a feel for what it's like to start a company. And we also have a formal lab-wide entrepreneurship class that was started at JBay. We also have customer discovery classes at Berkeley Lab. So on to some technologies. I've just selected a handful of technologies that are basically sort of portfolios. These are samples of our technology. At the end of my presentation, I have a link to the JBay site that has basically listed all of our technologies. And also the Berkeley Lab website also has a lot of other bio IP. One of the platforms that we're working on at JBay is polyketide synthases, which are enzymes that work like Legos. And this is our CEO's big project, Jay Kiesling. And one of the highlights of that was a technology that can produce diols, amino alcohols, and carboxylic acids and streptomyces. They've also started working in other host organisms. Some of these molecules are drop-ins. Some of them have never been made before, so we don't know what their characteristics are. That's one of the problems we have in deciding what to do at JBay. It's very hard to get information on what kind of new molecules might be interesting to industry. So we'd love your feedback on that. And it can be things, maybe you don't want to give away the secret sauce of exactly what the structure of the molecule is, but you can give us an idea of something that would be similar. So we always like to be in touch with industry about what the needs are. Of course, it's very hard to predict to structure to function. So if we create new molecules, you can't always predict what those functions are. We hope AI is going to improve that in the future. But anyway, in this case, we have both drop-ins and some new molecules. For instance, we've made 1,3-butane diol at 7 grams per liter. We're still improving the rate, but that's very early stage research with very little optimization. And that molecule is used as an emollient a lot of times in cosmetics. Another molecule that we've made at significant titer is tetramethylpyrazine. This is a coffee flavor and fragrance, and it's also being investigated for anti-inflammatory and antioxidant properties. And we're making that in C-glute at about 1 gram per liter. And that sound, they are low. We understand that for fragrances and flavorings, you may want to have 10 grams per liter or more, depending on the cost, the price that you can get. But we think it's a great start, and that's why I picked some of these out. We also have a whole portfolio on valorizing lignin, depolymerizing it using chelator-mediated Fenton reactions, and also depolymerizing and valorizing using metal catalysts. So there's lots of lignin in the world, and it's not a very expensive feedstock. So we really want to do some of these aromatics can be really valuable. We have a whole portfolio of what I call implanto production of chemicals, muconic acid, PDC, beta-keto adipate, arbutin, and gallate. We produce these directly in the plant. Sometimes it's in tobacco, sometimes it's in sorghum. We work a lot in that. We do also some initial work in Arabidopsis, which is, of course, not a commercial plant, but very useful model plant. These molecules can be extracted directly from the plant using hot water or methanol. And we have done some TEA to show that, for example, with muconic acid, if you can get 4% dry weight content of muconic acid in a plant, you can be better off techno-economically than doing a fermentation process. So we think this is a really interesting new, it's not new to the pharma industry, right? They do this molecular farming all the time, but for more commodity chemicals and things like monomers for polymers, things like that, it's not common. We have a lot of software that can be licensed just very easily, click licenses. One is our automated recommendation tool, which leverages machine learning and probabilistic modeling to guide synthetic biology. And you don't need to have a full mechanistic understanding of your biological system to be able to use ART. So basically, ART provides a set of recommended strains to be built in the next engineering cycle, along with probabilistic predictions of what their production levels will be. And we have, I think I listed a few companies earlier that have licensed this technology, this software, and found it very useful. So I'm happy to put you in touch with that. Our lead AI ML person at JBay developed that model. We also have very practical tools for TEA, LCA that can be licensed and leveraged. These have been developed by Corinne Scown's team, and very, very respected TEA, LCA researcher, been called on by DOE for setting various kinds of policies. And this is a very, very useful tool as well. And this may be less relevant to you because it's centered in the US, but basically a biociting tool for trying to figure out where you might build a fermentation facility, where there are resources, where sites that are close to infrastructure that you might need, grid, CO storage, and things like that. So another tool, maybe a little less relevant for you, unless you want to do US manufacturing, which would be awesome, and pretty much a requirement for JBay technologies. We also have a new downstream processing tool, which is, we've found that JBay has been a struggle to actually figure out what the cost of downstream processing would be. And we'd like to think more about that up front when we're developing our strains and selecting molecules to produce, because that can be a big part of the cost of producing a bioproduct. There are lots of other technologies at Berkeley Lab that are bio-related that I didn't have time to talk about today. We have a whole facility called the Agile Biofoundry, which is also a collaboration with DOE Labs. And this is for rapid design, build, test, learn for SynBio, all kinds of proteomics tools. ABPDU, which Deepti is going to talk about next, is part of the Agile Biofoundry. I've included a link when you get this presentation that you can click on for that. Or I guess I should put the URL because this will probably be a PDF. We also do plastic and mixed waste recycling, chemical and bio-based, critical minerals recovery, gas fermentation, soil plant microbiome research, and natural product screening and development. So I encourage you to look at not only the J-Base site for technologies, but also the IP office site, which I've also listed here. And that's it for me. So I think, are we going to save all the questions and answers for the end, or should I take Q&A now?
I think that we will save it to the end, since some of the questions addressed to you and some to Aviv, or Rami, so we'll address them at the end. But it was so interesting, and I think that we could speak about this for hours. So thank you very much, Robin.
Thank you for being interested.
And now I would like to welcome Dr. Deepti Tanjore, the Director of the Advanced Biofuels and Bioproducts Process Development Unit at Lorn's Berkeley National Lab, to provide us with an overview of the unit's operational model and collaboration opportunities. Deepti, thank you for joining us today, and please, the floor is yours.
Thank you so much, Ifat. I first of all want to thank the Israel Innovation Authority, Dr. Aviv, Tami, and Ifat. It really took a lot of work to get this webinar going. Really appreciate you being patient with us. So as has been mentioned a couple of times now, I am the director of a scale-up facility called the ABPDU. We have a little bit of a different model. What you have seen so far, what Robin has explained, is about the research work that goes on at Berkeley Lab, where you can license the patents for that technology and scale it up. At the ABPDU, what we do is we are more outward facing, as in we bring technologies both from JBay and other entities within the lab, but also from external entities like industry and academia internationally, and scale up their technology. So that's what I'm going to talk about for the next 20 minutes or so. So this is a view of ABPDU. We are on the 3rd floor of this building in Emeryville, California. The first 2 floors belong to a company called Amyris, which is a very popular company in synthetic biology. The 4th floor is where the Joint Bioenergy Institute exists. And the reason why I'm mentioning Emeryville is because Emeryville is a hub for synthetic biology, especially for industrial biotechnology applications. So if you are ever in the area, please do let us know. We'll be happy to give you tours and connect you with people here. So why do we even exist? So we are a Bioenergy Technologies Office funded entity within the US Department of Energy, EERE. This is a fisheye view of our fermentation suite. You see 2-liter bioreactors, and there's a 50-liter bioreactor in the front, and then 2 300-liter bioreactors in the back. We exist because our facility understands and resolves scale-up challenges for technologies that have been developed at the lab scale in milliliter scale. So typically, a lot of the synthetic biology or novel synthetic biology, or even general industrial biotech technologies are developed in well plates or even smaller volumes. But in the real world, this is how bioreactors look, especially with the lignocellulosic feedstocks that Robin mentioned earlier. These realities of the physical behaviors of these 2 vessels are completely different. You have capillary effects here in the milliliter well plates, whereas in the reactor, you have a lot of turbulence, heterogeneity, not just spatially, but also because we run bioreactors for longer time, microbes also tend to become heterogeneous over a period of time. They're not isogenic anymore. So there's this just completely different conditions. And this transition from milliliter to 2-liter scale has been challenging to many, many companies. Many companies have found that if they were able to transition this scale to tens of liters, hundreds of liters, it's actually possible to scale up much further, much better. But this transition has been what has been referred to as valley of death. And that's why Department of Energy funds us to be available to work with industry on a cost recovery basis. And I'll explain what that is in a moment, but be available as a resource for startups or small companies, big companies, and help them scale up their technologies. We like to say we meet our collaborators where they are. So if you're an academic entity, maybe you only have access to Shake Flask, so you can come to work with us there, and we will scale up all the way through amber systems at 250 ml scale, 2-liter Sartorius reactors to 300-liter bioreactors, which is our maximum capacity. But if you are a more mature company, you already have your 2-liter reactors, a process engineer who knows what they're doing, and they just want to run the 300-liter reactors, we're happy to do that as well. We also have upstream chemistry-based reactors for chemistry-based technology scale-up. For example, these are 10 ml tubes that people often use to study deconstruction, lignocellulosic biomass deconstruction. So we can start there and work our way through our pressure vessels at 10 liters, 50 liters, and 200 liters scale. We also have a dozen or so downstream unit operations. And when we don't have a unit operation, which often happens to be the case with downstream, we tend to rent, install, commission the equipment for our collaborators, and then decommission, send it back to our vendors. So all the italicized gray color font you see here was equipment that was rented and used for a particular project and returned back to the vendors. The vendors are very eager to be able to share their equipment, even their knowledge. They come on site and optimize the equipment for our collaborators because our current collaborators are their future customers. So we have that really good ecosystem going here at ABPDU. We have a dozen or so, again, analytical equipment pieces that help us analyze materials that we are generating, the samples we're generating through process development. However, if we need additional equipment, if there's a particular problem, a challenge that we are unable to resolve, then we leverage other facilities at Berkeley Lab, like the Molecular Foundry and Advanced Light Source. I would encourage you all to look up Molecular Foundry and Advanced Light Source because they also have their user facilities, and they have a proposal process, and they have a very interesting equipment and expertise worth looking into. So if we were working together, if you are a company or a university, we were working together, this is where we are right now. This is our initial contact. You're learning about the ABPDU, and then we proceed from there towards an NDA and project scoping. And project scoping will involve us coming up with all the tasks and subtasks to the resolution of 4 hours per subtask for the team members, for the industry members to realize where the costs are, what the scope is. And once we take the draft of the statement of work and we agree on it, then we will go into a more formal contracting processes, which will involve budgeting, contracting. Many of the contracts will go through all contracts go through our strategic partnerships office. And Robin already mentioned the different types of contracts that exist. And then we also pay a lot of attention to EHS and other kind of paperwork that needs to be done before a project can proceed. I do want to mention that our typical customer engagement is 12 weeks to initiate a project, but only when it is a US company, which is defined as all C-suite members and board members being US citizens. So in this particular case, it's important to note that if there are internationals, which is potentially the case with this audience, then we would have to go through international review, which could be 6 months or longer. So that's one thing to keep in mind. So it's better to get started sooner than waiting for later if there are international international founders who are international to the US. So this is, again, a process has overview of process hazard reviews that we look at. Safety is our highest priority, so we will really thoroughly review safety issues before we can start a project. And that's also actually a lot of knowledge that is useful for our collaborators later on when they're scaling up to commercial scale to understand where the hazards come from. Here's a quick example that I like to give. This is from a company called Hue. Hue was founded by Michelle Zhu and Tammy Su. Tammy was a graduate student at UC Berkeley, and she developed this technology to produce indigo via fermentation. Indigo production is a very toxic process. It's actually so toxic that we cannot do it in the US. It's done in other parts of the world. It involves using cyanide. So changing that chemistry-based process to a biology-based process and bringing manufacturing to the US and elsewhere was the goal of this team. They joined the accelerator program called IndieBio in San Francisco, and they needed enough indigo dye to stain a pair of jeans for their demo day when they were going to present it to their future investors. So they came to us and they wanted that amount of indigo, and not only to show it on demo day, but also to demonstrate that the indigo would behave the same way on an industrial machine like a traditional indigo would. And we were able to initiate this project within 6 weeks because we had a very tight timeline, and our strategic partnerships office came through and really helped us, and so did the IPO to get the NDAs and everything in place. And the dye you see here on this machine is actually the indigo dye that came out of ABPDU fermenters. And then there wasn't enough dye for a whole pair of jeans, but enough to showcase at the demo day. This is typically the story of many of the companies we work with. Two-people companies invent something during grad school or as a postdoc, come to us, we help scale up their technologies from milliliters to hundreds of liters scale. And while most of our collaborators are these small startups, we do work with the big companies. We work with the Shell, the Ginkgos, and the Rio Tintos of the world. And we also work a lot with academic collaborators. So as we are working with industry, we see industry-wide problems, and then we write proposals with our academic collaborators, solve those industry-wide problems that no one company wants to solve because there's no real return on investment. But once we've developed the tools, the methods, they're available for the public to benefit from. We also help startups raise non-dilutive funds through SBIR and STTR grants from multiple agencies, not just Department of Energy, but NIH, DARPA, USDA, NSF, what you have you. Apart from de-risking the technical process towards scale-up, some companies have also able to use the ABPDU's facility to test market risk. What I mean by that is they were able to produce enough product to test the market, as in will the market accept the product before they build their own pilot plant or commercial scale facility. So we've been able to help there and also help them do scale down experiments. When they go to commercial scale, if they have problems, they come back to us and we do scale down experiments to resolve those problems for them. So there are about 20 products in the market that were initially prototyped at the ABPDU. Ripple milk is a really good example. There are stores around the Berkeley area where you can go buy Ripple milk from the shelves in the store, and it was initially prototyped at the ABPDU. And what this does, this type of de-risking of technical problems and also market risk, helps these companies raise the equity value. And once they raise the value of their equity, they're able to raise more money from private financing, private equity funds. So I want you to focus in this inner circle here, because these were companies that fully relied on the ABPDU to produce their first prototype. And these companies have raised close to $3 billion. This is worth noting because the total investment that has gone into ABPDU so far is around $60 million, 61.5. So we're seeing a 40x return on investment, the value created here for private equity and private industry to gather from the investments into ABPDU. We conduct industry listening days. So these are open virtual events, and international audience are welcome. And we bring our Department of Energy colleagues. So here you see Mark Schmorhan, who attended this industry listening day before COVID. So we were all meeting in person back then. Now we're doing these more virtually. And we get a variety of different companies, different technologies, different backgrounds. People come together and give us a lot of feedback. And all that feedback merges really well into themes that we can pursue. So we're really here to help support industry. So we don't take any decisions of our own in terms of investment into equipment or resources without some industry input into it. A good example is before COVID, this was before COVID, we've received feedback that they wanted us to upload YouTube protocols on our YouTube videos on our operating protocols. So we developed these video SOPs that we put on our YouTube channels. If you go to YouTube and search for ABPDU, you'll find a lot of videos on how to operate large-scale equipment. And this was very helpful to the small companies that do come on site and work with us, but once they leave, they don't have any materials to look at. Now they can just go to YouTube and they'll find all the materials they need to kind of realize what the scale-up challenges are for them and solve those problems independently also. Another example of a theme that came up in 2019 was resources for gas fermentation. So we were asked by the industry community to invest in process development scale gas fermentation, that is 0.5 liter and 20 liter bioreactors. So here you see we bought these HEL bioreactors from UK, which are pressurizable. They can go up to 6 bar pressure. And we set them up for 5 different gases. So the manifold is pretty intense here because we can do syngas, we can do methane, we can do carbon dioxide, of course, carbon monoxide, etc. So this is important to note because this investment was close to $1 million on our end. But we've been working with over 6 small companies right now scaling up their technologies. In the past, these companies had either access to a serum bottle at lab scale, or then they have to just go to commercial scale, and there was no intermediate scale they had access to, and now we are able to offer that to them. If you are a small company, this is a huge challenge, equipment and equipment access, and that's what we continue to offer for the past 15 years, and not just equipment, but also expertise. I really particularly like this equipment because this also doubles up as pressurizable liquid state fermentation equipment. In this case, this company called Checkerspot was able to use this equipment to pressurize their fermentation process to be able to simulate how the pressurized reactions behave, or pressurized cultures behave in a commercial scale reactor. So if you think about a 100,000 liter or 200,000 liter commercial scale industrial biotech reactor, the bottom layers experience a lot of pressure from the weight of the water column on the top. And as a engineer, it's important that we figure out what that weight does to the culture and have that information before scaling up. And we are now able to offer that to our collaborators. I'm going to wrap up in a few minutes here, but before I do that, I quickly want to point out to a particular concept that we are developing called the science of scale up. The idea that scaling up and biomanufacturing at scale is mature is actually not true. In fact, there's a lot of basic science and some applied science problems to be solved at scale. Often we think that basic science problems are only at the molecular level. It's actually, there are molecular level phenomena occurring at scale that need to be resolved at scale. So that's something we've been looking at since 2014. We included it in our strategy. It ended up in Biden-Harris executive order in 2022. At LVNL led this theme for a series of workshops with Bioenergy Technologies Office in 2024. And the National Security Commission on Emerging Biotechnology put this recommendation in their report, and it suggested $500 million investment earlier this year. Over $175 million of this recommendation was recommended towards DOE for improvement on process technology and equipment, especially hardware, software, and digital signal processing. So I just want to mention this because, as I mentioned earlier, we see industry-wide issues and then we try to go after them when no one company is looking to solve them. One example of that is my personal research interest on self-driving bioreactors. So this is the state-of-art bioreactor operation right now. We conduct fermentations. We collect real-time data, collect offline data, then we analyze it off-cycle, and we learn something from it. So if you're a junior engineer, you probably don't have a lot of experience in intuition. So when something is happening with a bioreactor, you're probably just watching it occur. So if it's foaming over, or you think it's about to foam, or you let it foam over until you put before you add anti-foam. But if you're a senior engineer, you probably think, oh, I should intervene and I will put anti-foam sooner before it foams over. This kind of decision making and heterogeneity associated with that decision making often leads to people thinking of scale up as more of an art than science. And the best way to resolve it is automating some of these approaches. The first thing we are trying to do right now is actually develop novel real-time data that can help us analyze the system better. And we are looking at imaging, quantum sensing, and a few number of Raman and other technologies to be able to provide good recommendations to the process engineer. So it doesn't matter whether they're a junior engineer or senior engineer, they're getting the same kind of recommendations through the application of AI on this novel real-time data. We're also looking at doing this enough number of times and coming up with the rules of microbial heterogeneity as it occurs in bioreactor that we can optimize, develop an optimization algorithm. So now it's a completely self-driving bioreactor. That's maybe a little bit further out. Right now we're focused on the real-time data. And an example of the real-time data that we've been developing is this collaboration I have with the Ajoy group at UC Berkeley. They are quantum sensing experts, and they've helped develop quantum sensing for intracellular data from bioreactors. In the past, the way quantum sensing with nanodiamonds was being done is more with a lateral flow assay, like a pregnancy test, and that is incompatible with a bioreactor operation. So we had to transition that technology into a droplet-based method. So now we can continuously draw out of a bioreactor, which is one good thing about bioreactors. You can do some destructive sampling. So we can actually draw out milliliters per hour and understand what's going on intracellularly in the cells in the bioreactor. This paper was published last year. So that's about what I have, and I want to thank all the people who do the actual work, and I'll stop there so we have a few minutes for Q&A.
Thank you so much, Deepti. That was a very insightful presentation. We received a few questions, and Aviv already answered to some of them in writing, but we have a couple of questions to you, Robin and Deepti. Robin, we have a question. Are microalgae suitable for JBEC collaboration?
Yeah, microalgae is not something that we're funded by the Department of Energy to work on, but we do work on microalgae. We have people that do that, and some of them are at Sandia. They're not at Berkeley Lab, but yeah, and it's not a major focus, but we absolutely can do it depending on what the work is. And we can also help you connect with other people in the National Lab system that work on microalgae or macroalgae, both.
All right, thank you. The person that asked that, if you need, we can help with some connections after the webinar. Another question is, I think to you both. Do you have other collaborative departments for solar cells or other types of energy?
Yes, we actually work on early stage perovskites and all other kinds of solar materials. So basically new types of solar. We don't work on like structural engineering problems with solar, but we work on materials. We have I think Deepti mentioned the molecular foundry, and we have a huge material sciences area with actually a lot of UC Berkeley professors are joint appointees with Berkeley Lab, and yeah, so lots of new solar research. We also have actually something called Flex Lab, where you can actually bring new materials, new building types of materials, window coatings, different solar types of technologies, depending on what they are. Not huge solar, but smaller, more localized solar, that we can actually test those materials. So we have really early stage, and then we have, fairly well-developed facilities.
Okay, thank you. And if I recall correctly, NREL has a significant research
Yes,
department.
they do. Absolutely,
Yeah.
yeah, they do.
Okay, and again, if needed, please contact us and we will help with the connection. We are reaching, I see that we have some more. Are there projects dealing with biosensors?
Yes, we do have projects dealing with biosensors. I don't particularly look at them, but there are other PIs that I'm happy to connect you with. I also realized I forgot to go over my contact information. You can reach me at abpdu@lbl.gov. So it's just the name of the facility, abpdu, or my personal email ID, which is D, my first name initial, and last name Tanjore@lbl.gov. Either ways, I should be able to respond within 24 hours. So feel free to reach out to me.
And biosensors, there's also Los Alamos has some people working on biosensors and argon, and they work through the Agile Biofoundry, which is another DOE consortium that I just mentioned in my talk. So we could put you in touch with Terica Dale. Works on depending on what you're trying to sense and what they're for. But between Deepti and I think we could probably find somebody to refer you to.
We will send to the participants of this, to the audience of this webinar, some information. After the webinar, we can send the presentations with some contact information, so you can email to each one of us to receive more information or relevant connections. Is it possible to collaborate in area of recombinant proteins?
Absolutely, yes. We routinely make recombinant proteins for our collaborators. We're happy to scale up, and I'm pretty sure Robin will say that JB will actually happy to help with engineering as well. Robin?
Yes, absolutely. Yeah.
All right. So we've reached to the end of this webinar, and once again, we will send to you all some more information and contact information so we can continue communication. I would like to thank again Deepti and Robin for joining us today, and thank you Tami, Aviv, and Rami. We wish you all best of luck. And yes, we will share the recording as well.
Thank you so much for the gracious
Thank you so
invitation.
much, Ifat.